So, Professor Jon Deeks, Deputy Director of the Birmingham Clinical Trials Unit, which is where we're based, the senior methodologist on the trial, is going to talk to us and summarise the negotiations we had with NIHR around the sample size for BASIL-2, and he's promised not to baffle us with too many figures. So, thanks, Jon.
What am I clicking?
Just the arrows. Yeah.
Good morning, everybody, and it's a pleasure to talk to you. Having worked on this trial behind the scenes as a methodologist and never actually encountered anybody other than the immediate Birmingham team, it's great to see so many faces who are involved in running this trial. So how many participants do we need now in BASIL-2? We started off with wanting 600. That was the target sample size in the trial. And I'm just going to briefly explain where that number comes from and why it is we can actually get away with slightly less than that now. So the whole point of a sample size calculation is making sure when we do the study and we look at the data at the end, we can distinguish what is a real difference from what is just a difference which is there due to chance. We all know the chance plays tricks on us, and that when we actually look at things, we have to be clear as to what are the sorts of differences we see which are there just because that's what's happened, and which ones actually are large enough to be clear that there really is a difference. So the sample size is the driver for actually doing this. It determines the precision with which we know the result of our trial. In our case, we're looking at the estimate of treatment benefit, and we want to know how precise that is. And we need to ensure that the estimate we will get at the end of the trial is precise enough for us to be able to separate out the chance effects from the real effects, so that anything which is large enough to be important, we can be clear is a real effect. So in BASIL-2 and BASIL-3 as well, the primary outcome is amputation or death. There are secondary outcomes, but this is what we're really focusing on. And our analysis, which we will do at the end of this, considers both the incidence of this outcome, but also the time to it occurring. Everybody will die at some point, so it's actually a lot of what we are hoping to do is to see an extended survival time, an extended time of good health. And we have, because we're looking at both the time to an event and the occurrence of the event, we actually can take into account we can gain some information from that analysis. We compare across all the follow-up time that we have accrued for all our patients. And when we set up the trials, we actually set them up to be able to detect what we call a hazard ratio of 2/3. So this is actually based on thinking about we could see a difference if it was as large as this difference here of 34 versus 47% having the event at 3 years. If we wanted to see much smaller differences, we'd have to do a much bigger trial. So that's our decisions, and this has all been based on the experience from the original BASIL trial, which is now known as BASIL-1, although it was never called that during those days, but we'll call it BASIL-1 through the rest of the day. So that's the magnitude of effect we want to see. Now, in doing the sample size, there's some statistical equations which get thrown around, but they're really doing two things, and they're controlling our statistical error rates. And we can make two different errors when we do a trial. One is to say things are different when they're not, and we call that a type 1 error. Not a very useful name, but there you go. And the way we control that is our p-value, which we use in the analysis, and we've set that in our protocol to be 0.05. So we're saying there's a 5% chance that at the end of the day we might be wrongly concluding, and that's an industry standard, if you like. The other error, surprise, is known as a type 2 error, is when we miss a difference which really exists. So that would be wrongly concluding the treatments are not different by at least the magnitude we've defined when we're actually seeing a non-significant result. And that is driven by the sample size, and we increase the sample size until we can get the mathematics to tell us what the probability is of making that error. And so we've set up BASIL-2 to have a 90% chance of detecting a difference if it is there. So that's the statistical power of the study. Now, we're not changing these issues. We're not changing them at all. What we're actually have in a trial like this is that what defines those probabilities is the number of events we see. It's not the number of participants, it's the number of events. And to see this magnitude of difference with this control of statistical errors requires 247 events. So one option we have is actually to stop recruiting now and follow everybody until 247 of them have had these events. We don't want to do that because it will be a long time, and we want to get an answer for this question sooner than that. So we want to increase, we're going to have more participants. But the two things in our favour now, not in the favour of the participants. One is that actually we are seeing different event rates than we forecast. And secondly, we are actually taking longer to recruit, which means we've got longer time periods of participants in follow-up. And so we will be seeing more events because we are following them for longer than we originally thought. So originally it was going to be 3.3 years would be the median follow-up, and because of the slow recruitment, we're going beyond that. Let's just have a look at the event rates first. So we have data now on events in the first 2 years of follow-up for some participants. What we see in this table is on the left-hand side, the BASIL-1 are the values which we use in the sample size calculation. So in year one, by the end of year one, we expected to have 24%, which is why we've got 0.76, having an event by the end of year one. And we've actually seen 26%. So these figures in the table are the survival probability. So it's the proportion not having an event. And so the year one experience was very similar to BASIL-1. But year two has been a lot higher event rate. In year two in BASIL-1, which is what we use as our basis, there was only going to be an extra 8% of participants having events. What we've actually seen is a lot more than that. It's, 14, 15, 16, 17%. So there's a lot more events. That means that we've actually got more outcomes occurring. Matt earlier on showed you data from the BASIL-1 subgroup, which I'm showing you here. Our current event rates are the red line on this slide, and the blue line at the top is the BASIL-1 subgroup. So you can see we are actually have more events even than that selected group. The green line is the series from the Heart of England Foundation Trust. So our events are much closer to what they are seeing there. But we don't know what's going to happen in years 3, 4, 5 of follow-up. We haven't got that data. We can only hypothesize about that. So how many participants do we now need? Well, if we went with our original figures of event rates from BASIL-1, which we now know are conservative, because we've got longer follow-up, because we've recruited more slowly, we would expect to need 505 participants to be able to see 247 events. So we've gone down from 600 to 505. So the gain there is because of longer follow-up. That was based on us continuing to recruit at 6 per month, and it would mean that we'd keep on recruiting into this study through until April 2021. And the follow-up would then end 2 years later in April 2023. So that's quite a long time to go. If our event rates that we're seeing now in year 2 of BASIL-2 continue, we can actually get away with less patients. We'd only need 415 patients to be able to see our 247 events. And that means we could, if we keep on at 6 per month, we would recruit until January 2020. So we can knock over a year off the study if those event rates continue. If we increase our recruitment rates above 6, then of course we can finish this study more quickly and answer this important question sooner. So in summary, BASIL-2 will be delivered with less participants than the 600. We don't need it because we only need 247 events, and it will be unethical for us to continue the study when we already will have the answer at that point. The number we actually require will probably sit between those two numbers of 415 and 505. We don't know what it is yet. It's going to be dependent on what the event rates are in years 3, 4, 5 of follow-up for the participants. We've put in place a robust decision-making process that decision as to when to stop will be made by our independent trial steering committee based on the data that we observe in the study. So that's going to be done in a scientifically credible way at the right point in time. And they are basically going to be reviewing what's happening every 6 months until we get to that point. But this requires six participants each and every month to be recruited, and more than 6 would, of course, mean that we can get to the end a lot sooner. So thank you very much.